Welcome to Lindamood-Bell Radio, a podcast where we explore topics in literacy and learning. I'm your host, Dave Hungerford, Lindamood-Bell's Director of Visual Media. Today I'm talking with Associate Professor at the University of Alabama, Birmingham, Co-Director of Undergraduate Neuroscience Program, and the Director of the Cognition, Brain, and Autism Laboratory, Dr. Rajesh Kana. Dr. Kana, welcome to Lindamood-Bell Radio.
Thank you. Thank you.
Tell me a little bit about your laboratory and your research interests, just to get us started today.
So my laboratory is called the Cognition, Brain, and Autism Lab, which tells you that we are interested in examining different aspects of autism spectrum disorders. So we primarily study the brain in autism from different points of views. By that, what I mean is we use different types of neuroimaging techniques to understand function, structure, connectivity, and even chemical concentration in the brains of children as well as adults with autism.
And what are some of those techniques and procedures? I know the fMRI is a big
That's
part
right.
of it.
Yeah, fMRI is one of the primary techniques which we use, which, as you know, stands for functional magnetic resonance imaging. So we can have a person go into the MRI scanner. We ask them to read a bunch of sentences in the scanner or view a movie clipping, and then we look at how their brain responds to those sort of events. That's one technique called functional MRI. We also use a technique called structural MRI, which is you don't have to watch anything. You just lie down in the scanner static, and then we take photographs of the brain. By using that, you can measure the volume, thickness, surface area, etc. of different structures of the brain. And occasionally we use techniques like diffusion tensor imaging, and that's a technique which helps us understand the white matter connectivity of the brain, how axons are connecting different neurons in the brain and pass information from one place to another.
Well, over the past couple decades, it has been widely publicized about the increase in the number of autism diagnoses. Is there some sort of consensus or road that we're going down that is starting to find some results on either prevention or accommodation for individuals that have that diagnosis?
Yeah, I think prevention may be a difficult thing to achieve, probably because it's a disorder and which has more biological basis to it, and it may be complicated by the environmental differences. Also, it's kind of like a brain behavior sort of cycle triggered by biological differences, and interestingly, to reduce the impact of the disorder on the behavior and the life of a person with autism, there has been a lot of different types of treatment and training procedures, and therapeutic ones, changing the behavior, changing the problems in different aspects of behavior and thinking and so on and so forth.
Has there been some sort of consensus as to the triggers? Is it environmental? Is it genetic? Is it a little bit of both?
I think, so far we only have scientific evidence to support the biological basis of the disorder. So there are several different genes implicated in the disorder. And in some ways that makes it difficult also to box in which gene is responsible for autism, because there are plenty of genes responsible for autism, and that kind of changes the brain development, which affects the behavior. So it's kind of like a cascade of different processes affecting and creating this syndrome.
Well, one thing I did not know is that you were co-director of the undergrad neuroscience program there. What does all does that entail?
Huh. So the undergraduate neuroscience program is a collaborative program. It's what we call an interdisciplinary program between two different schools, the School of Medicine and the College of Arts and Sciences. And within those two different domains, we have one department, neurobiology, underneath the School of Medicine, and psychology underneath the College of Arts and Sciences. So we collaborated to create this program where students can get exposure in neuroscience at the undergrad level itself. So the students enrolled in this program take courses from biology, physics, math, chemistry, neurobiology, and psychology. So they get a good basis of the fundamentals of neuroscience before they branch out to go to higher studies, either to med school or PhD programs or anything like that. So it's a very effective, that's what we have found out, interdisciplinary program.
And you have a large medical center there. I've been to the campus.
We do, yeah, we do, and I think that's what helps this sort of program to be successful, because one of the things we do in the program is basically have every single student do a research project as part of their undergraduate 4 years of program. So that makes them work in a research lab, either in the School of Medicine or in the College of Arts and Sciences on a neuroscience project. So that gives them a lot of training and skill development before they make a decision to go for their higher studies.
That's great. They get some practical experience before
they do
graduate school.
Mm-hmm.
Well, we have worked together in the past, and one of the interests in Nancy Bell's work has always been figuring out how to help students who can read or decode higher than they comprehend. And our learning centers have always had students with this kind of profile. And a few years ago, we were fortunate enough to work with you, like I said, and provide some students for a study that was rather involved students that had the autism diagnosis. Maybe you could talk about what the impetus for that was and kind of describe that project, if you will.
Mm-hmm. So yeah, I think the Lindamood-Bell project, the collaborative project, was one of the main projects which we have done as a lab in the past few years. And I think it came about from one of my publications in 2006, which examined visual imagery processing in individuals with autism and how their brain responds to different levels of imagery. So in that study, we had sentences like number 8, when rotated 90 degree, looks like a pair of eyeglasses. So that's a sentence to process that you have to imagine and perform mental rotation, basically. So how people with autism respond to such kind of challenges and how their brain activates or do not activate to such challenges. That's what that study talked about, and I think that fitted really well with Lindamood-Bell's basic premise in terms of approaching intervention related to reading in dyslexia as well as in children with autism, which makes them think in terms of visual imagery or building a visual imagery to tackle the problems in language comprehension. I think that was the place where the two different sections came together. But by that, what I mean is, our lab as well as Lindamood-Bell came together to do this collaborative project.
And so we had students that I recall, we'd hoped it would be local at first, have students come in that had a certain criteria that they could decode at a certain level, read at a certain level. They comprehended less than that. They had a diagnosis of being on the spectrum. Mm-hmm. What was the process that they went through at the lab?
So exactly what you said, like they had to have a average reading level but below average comprehension. That was one of the criteria to be included in the study. And once we screened the participants, they come to Birmingham, to UAB, to our lab, and then we had a lot of testing as one part, in about 2.5 to 3 hours of neuropsychological testing, where we measure IQ, language processing, lots of different tests to understand language and cognitive aspects. And then they go through an MRI scan for about an hour. So they spend 60 minutes in the MRI scanner doing structural scans. Also, in addition to that, we had different types of functional scans where they solve different types of language problems, word processing, sentence processing, narrative sort of things inside the scanner. And then we look at their brain activity.
And these are children.
These are children. These are children who are between 7 years and 13 years.
And how do you get them to sit still in a scanner for an hour?
Yeah, that's probably one of the biggest challenges, and we had all different kinds of experience as we were going through that process. But I think we developed a very clear plan for our scan, which is basically once they come in, we familiarize them with the scanner. Before they came in, we send them social stories about how their day is going to go about, which means we send them picture stories that you're going to come to this building, picture of that, you're going to go to this machine, which is a scanner, etc., etc. That's one part. The second part is, we send them in advance the scanner noise on a CD so that they can listen to that noise and acclimatize to the noise of the scanner, which is relatively loud. The third one is when they get here, we took them to the scanner and we pretty much booked 15 minutes of actual scan time, kind of as a mock scanning, but the real scanner. We put them in the scanner, and that 15 minutes we don't scan, but we create the noise, everything is there, and they get that experience of being in the scanner for that much time. So all these different steps help them get used to the whole process of scanning environment, and then they come on a different day to get into the actual scan. I think that sort of approach has helped significantly improve our data quality. Having said that, that's not 100. I mean, none of these processes is 100%. Foolproof in the sense that every child has different sort of needs in autism, and the younger the child, it becomes more and more difficult. But I think overall, it gave us pretty good yield.
Yeah, I seem to recall that it was quite successful. Actually, only had one or two that were completely unusable, which.
That's right. And imagine that, they have to do one scan and then go to their intervention and come back for the second scan. So they had to go through this twice, pretty much.
That's right. And we had students that we had identified at different learning centers of ours all across the country who flew to Birmingham and took part in this.
That's right.
Received about 10 weeks of visualizing and verbalizing instruction, which, our kind of the basis of that program and our efforts is to work to develop that the student can visualize language and make very concrete imagery in their head that matches that language, be able to verbalize that. They would come back after 10 weeks, take behavioral tests, the fMRI. Comparative, we had students who also fit the criteria, who served as a control group, who did not have the instruction in between.
That's right.
They received it later. What, in general, I know there have been several papers that have come out of this. What were the results?
Yeah, so going back to the point you mentioned, I think it was amazing to see that we were getting participants all across the country. We had participants, several flying from California to Birmingham, Philadelphia, New Jersey, Florida, Texas, all these places. They come to Birmingham to do this twice, and then after the first scan, as you mentioned, they go for this 10 weeks intervention, which is also very intense and massive. And I think, that's one of the criteria for a successful intervention, how detailed and intense that intervention is. So coming back to the results of the study, I think we published 4 papers looking at different things, but the basic aspect of the results, or one of the common themes of the results, is basically that behaviorally there was an improvement in their language comprehension, and the beauty of it is that improvement predicted their brain level changes. Especially one of the things which we found is there was an increase in connectivity between the two classic language areas of the brain, which we call Broca's area and Wernicke's area. So the connectivity between those two regions, which are traditionally involved in language production and integration and comprehension. So those two regions' connectivity improved, and that connectivity improvement predicted their language or reading comprehension improvement. So that was one of the common themes, and then we had subtle differences, but most of the changes in the positive direction from point 1 to point B after intervention.
Wow. And for those that don't know, the sort of vetting of your work when it goes into publication is very robust and thorough.
It is, yeah. So we the papers which we published are in really a good impact factor peer-reviewed journals, and I'll give you a couple of examples. One is Human Brain Mapping. That's a really good journal, and as you can see, it publishes most papers with neuroimaging. So the papers are reviewed by peers who work in the field, who we don't know. It's selected by the editors of that journal. Another paper was published in Autism Research, which is a leading journal in the field of autism, again reviewed by experts in that field, and we submit the paper, they review. We don't know who the reviewers are. 3 or 4 reviewers will review the paper, send it back to the editor, and the editor can make a decision whether to reject the paper or ask the authors to make changes to it. So usually what they did in these papers is send it back to us, and then we address the reviewers' comments, send it back to the editor sends it back to the reviewers again, and they give the comments again. So it goes through a couple of cycles, before the editor can make a final decision that to accept these things. So it's a very thorough process before it gets to the final product.
And it's a long process. Right?
It is a long process too, yeah. We are looking at minimum of 6 months, and if you're lucky, if you can get published in 3-4 months, yeah.
And so, as a researcher, you must be juggling at any one time several projects.
That is right, yeah. So we are, when you manage a lab and you're doing multiple different projects. So this was one of the main projects in the lab at that time, but at the same time, we were working on several other projects too. So in addition to my own role in teaching students, undergrad students, and the administrative work of directing the undergraduate neuroscience program, et cetera, et cetera. So we were working on several other projects where we were looking at social cognition in autism or individuals with autism. We were looking at structural differences in the brains of individuals with autism. So all these are going in parallel or integratively. But I think, the convergence of it is that, with all these studies, we are targeting or we are studying to better understand autism and to learn about how we can better treat that disorder.
What projects are you currently working on?
So 2 projects which we are, let me say, there are 3 different projects which are lined up. One of the projects is another study checking the effect of intervention on brain functions and structure in autism. And in this case, we are looking at an intervention called PEERS, PEERS. This is an intervention designed at UCLA, and it's basically teaching adolescents and young adults with autism social interaction or the skills of social interaction. So anything which they have difficulty in, like the etiquette of a dinner conversation or a party conversation or dating or anything for that matter, the daily life struggles a person with autism might go through, they can go through this intervention called PEERS. So what we are planning to do is to identify a group of young adults and adolescents and scan them at time point 1, and then they go to this 14-week intervention on social skill development, and then they come back for the second scan. And so we are in the design of that study at this point. The second study which we are going to be involved in is a study on driving. This is an interesting domain. We are looking at adults with driving license, with autism, and many of them, despite having driving license, don't drive, and that's because of various reasons. So their cognitive and social problems can be a major factor in their driving. So what we are trying to do is basically look at their performance in a driving simulator. So we have a driving simulator at the transportation lab at UAB, and we wanted to look at their driving there and also adapt that driving simulation into the MRI scanner. And what we are trying to look at specifically is while they're driving in the scanner, we planned hazards in terms of driving. Like, can they anticipate a potential hazard? There's a car coming, whether that car turns left or right, and do you need to adjust your speed? And or a pedestrian trying to cross. So we plan these and hazards which they can anticipate and how their response to that is going to be. The third project is what we are waiting for funding from NIH, and that is based on the Lindamood-Bell data which we collected. So the preliminary data which we collected from Lindamood-Bell, I used that to submit an R01 application, which is a big grant, and that went through second stage of review, and we are keeping our fingers crossed for funding at this point.
And what will that examine?
So they have scored a grant, so what that means is if you get a good score in that grant, my grant scored, pretty good, and they have a council meeting in May of this year. So next month they meet and they'll make a decision up to what percentile they would fund, and if we get that funding, we're going to do a lot of children. We're going to scan a lot of children, and then they will go through the Lindamood-Bell intervention. So it's kind of like a bigger scale project of the Lindamood-Bell project, which we earlier talked about.
And that's important to replicate findings from one project to another, correct?
Exactly, it is. So one level of replication would be that in the earlier project which we did with Lindamood-Bell, we had about 25 subjects in each group, and that's still significant for an intervention study that has two different parts to it. In this one, that number is going to jump up to about 100 subjects. So it's a large number of participants, and we also have a control group which is going to go through this intervention as well. So that replication is going to be huge, to understand the reliability and validity of our findings, which we got from our earlier study.
Got some exciting stuff going on in the background there, I can hear.
Oh, yeah. It's near the hospital, so there's always something or the other, yeah.
Well, where can people, again, read your work and maybe follow up on your research if they're interested?
So our lab website is www.uab.edu//CBRA, CBRA, and that stands for Cognition, Brain, and Autism Lab, CBRA. So on that website, I think we have details about the setup of the lab, the projects which we have, and the personnel working, and so on. People can look at the psychology department website, and also our work is published. So all the journal articles are available. Most of them open access. Others, if anybody needs, they can also contact us to provide those papers as well.
Great. Well, there's also a short documentary on the project on the Lindamood-Bell Learning Processes YouTube page, and it's titled Improving Comprehension for Students with Autism. It includes Dr. Kana's remarks and sort of walks us through the project, as well as interviews with families and even some of the students themselves that were involved. And you can, again, check that on the YouTube page.
Yeah, that's an excellent one, and I would totally recommend that anybody wants to know about the project as well as the collaboration with Lindamood-Bell and the whole project's backdrop. I think it's a very nicely made one. One thing I would say is, all our long-term goal in research is basically to help children and adults with autism and their families. And to achieve that goal, my lab as well as many other labs probably are trying to do translational work more and more. By that, what I mean is basically one level, we want to understand the mechanisms at the brain where we can point out that this may be a signature or marker for autism. And the advantage of that is that helps us probably take it to the next level of diagnosing a child with autism much earlier than it is right now, actually. And that helps in intervention, which is the second direction, which means, if any family comes and asks us that, okay, my child's brain behaves differently or it responds differently, what can we do about it? So the second goal is basically to understand these brain circuitry which are different and how the interventions like Lindamood-Bell intervention or any other probably can change that faulty circuitry so that the behavior or function can be improved, actually. So what we call in research, it's very much translational. That's the direction to help these individuals and families. And that's exactly they are looking for as well, I believe.
Well, you're certainly looking at a lot of very interesting facets of human behavior there.
Thank you for having me. Thank you.
Thank you for listening to the Lindamood-Bell radio podcast. I hope you listen to more episodes, and if you enjoyed this, please subscribe. Follow Lindamood-Bell on Instagram, Facebook, and Twitter @Lindamood-Bell.